Hello there. Welcome back to Midnight Ocean. I'm Velen, your guide to traveling through the wonders of the ocean. Tonight, we're going to discover some of the longest living marine creatures on the planet. If these cozy moments calm you, I invite you to subscribe, share a comment, or leave a like. Now, get comfortable, take a deep breath. Don't be afraid, you're safe here. Let's dive deep into the sea and let our imagination guide us to a peaceful and deep night. The glass sponge species Monoraphis chuni is recognized as the longest-lived animal on Earth, with age estimates exceeding 11,000 years. This sponge lives at the bottom of the Indo-Pacific, anchored to the sediment by a single silica spicule that can reach 3 meters in length. It tops a list of marine creatures that defy any human notion of time. The Ming quahog clam, accidentally killed in a lab after 507 years. The bowhead whale, which carried 19th century harpoons embedded in its own body for over 100 years and may live up to 512. The red sea urchin, which reproduces at 200 years at the same rate as a young one. The Hawaiian gold coral, with colonies dated at 2,742 years. The Greenland shark, which takes 150 years to reach sexual maturity. And Asterisk turritopsis. Dornii asterisk, a 4.5 millimeter jellyfish capable of restarting its own life cycle indefinitely. Isotopic analyses along the spicule of Monoraphis chuni show that the animal already existed during the last ice age, when the surrounding water temperature was 1.9 degrees Celsius. Each layer of silica added to the spicule is a chemical record of the year it grew. The oldest sponge on the planet is also a climate archive spanning the entirety of recorded human history. Think about the scale of what's being said here. While entire civilizations were born, flourished, and disappeared, a sponge anchored at the bottom of the ocean kept growing, millimeter by millimeter, without moving, without aging in the conventional sense. The glass sponge has no brain, no nervous system, no complex organs. Its body is made up of specialized cells that filter microparticles from the water and extract dissolved silica to build its spicule. A needle of biological glass that grows so slowly that measuring its progress in decades would be almost pointless. When the last ice age was coming to an end and the first humans were beginning to cultivate grain in the Middle East, this organism already existed. It hadn't just started growing that week. It was already there. To understand why this is biologically remarkable, you need to understand what aging actually is. In most animals, cells divide, accumulate genetic errors over those divisions, and eventually stop functioning correctly. That's cellular aging. Monoraphis chuni escapes that cycle for reasons that still puzzle researchers. An extremely slow metabolism, the absence of predators in the ocean abyss, a constant temperature near freezing, and a body structure based on silica, not vulnerable soft tissue, that doesn't undergo the typical degradation of organic materials. The extreme cold of the ocean floor acts as a permanent metabolic slowdown chamber, reducing cellular to nearly imperceptible rates. The silica spicule is not just structural support. It is simultaneously the largest silica object ever built by an animal anywhere on Earth, and a climate archive of extraordinary precision. Each microscopic layer deposited around that central axis contains specific ratios of magnesium, calcium, and oxygen isotopes that reflect the temperature and chemical composition of the water in that specific year. Think of it as a book where each page equals a decade, written in chemical language instead of letters. Scientists at the Max Planck Institute for Chemistry used geochemical analyses of this spicule to reconstruct deep ocean temperature variations over millennia, identifying warming episodes caused by submarine volcanic activity that no other record had captured. Life in the ocean abyss where this sponge lives is radically different from any land environment. pressure equivalent to hundreds of atmospheres compresses anything that isn't adapted to it. Sunlight doesn't penetrate beyond the first 200 meters. Below that, total darkness reigns. Temperatures stabilize near 0 degrees. Currents are slow and predictable. There are no seasons, no hurricanes, no sudden temperature swings. This extremely stable environment is, paradoxically, what allows for extreme longevity. Less disruption means less accumulated damage. The sponge doesn't need to flee, hunt, regulate its body temperature, or compete for territory. It filters, grows, and persists. Other animals on the list share similar longevity strategies, even if through different mechanisms. The Ming clam had a slow metabolism and reduced oxidative stress. The bowhead whale has specialized DNA repair proteins. The Greenland shark grows only 1 centimeter per year and takes over a century to mature. The jellyfish Turritopsis dohrnii reverts to its larval stage when stressed, restarting the cycle. All of them converge on the same fundamental finding. Extreme longevity is not an accident. It is the product of precise biological adaptations that reduce accumulated damage, repair genetic errors with unusual efficiency, or simply slow all vital processes down to the point where aging becomes almost irrelevant. The fact that Monoraphis chuni already existed when the water temperature around it was 1.9 degrees Celsius, a measurement recorded in the spicule's own structure, carries scientific implications that go beyond biology. That specific temperature was inferred from the ratio of oxygen isotopes deposited in the innermost layers of the spicule, the oldest ones. This means the sponge's body contains climate information from the period immediately following the last glaciation, data that oceanographers would normally need to extract from marine sediments or ice cores. A living sponge functions as a chemical witness to the planet during times that predate any documented civilization. There's something unsettling about realizing that this organism has no awareness of its own age. It doesn't accumulate memories, doesn't process experiences, doesn't feel the weight of the millennia. Its existence is purely functional. Filter water, extract particles, deposit silica, grow. And yet it is the oldest living animal on the planet. Extreme longevity, in this case, requires no neurological sophistication or behavioral complexity. It only requires a metabolism compatible with its environment, the absence of evolutionary pressures that favor rapid aging, and a body structure sturdy enough to last when everything around it changes and disappears. The discovery that specimens of Monoraphis chuni can live more than 11,000 years repositions the very definition of what counts as an animal. Most people associate animals with mobility, behavior, even feeling. Here, the definition is purely taxonomic. Multicellular heterotrophic organisms that take in nutrients from their environment. The sponge fits that category, even motionless, even without nervous tissue, even made up of a structure that looks more like a mineral than something biological. Coming to terms with that means confronting the limits of the categories we use to organize nature, and realizing that nature frequently ignores those categories with complete indifference. When researchers return to collect data from this spicule in the coming decades, they will find recent layers recording ocean temperatures during the 20th and early 21st centuries, a period of documented climate change. The same sponge that recorded the end of the Ice Age will record contemporary ocean warming, adding more pages to a book that began before human writing existed. Every new millimeter of silica deposited is simultaneously evidence that this organism is still alive and a permanent record of the planet's conditions at that moment. The glass sponge didn't just survive history, it stores it. The oldest non-colonial animal ever found was a quahog clam named Ming, dredged from Icelandic waters by researchers in 2006. By analyzing the growth rings on its shell, the same way you count the rings of a tree, Researchers discovered that this ancient bivalve was 507 years old. Ming was born in 1499, when Leonardo da Vinci was still painting. It survived all the European wars of the 16th through 20th centuries, the Industrial Revolution, and the rise of the Internet. Unfortunately, Ming was frozen and preserved in the name of science, something researchers regret, and we still don't know the maximum age these organisms can reach. North Atlantic quahogs show negligible senescence. They display no typical signs of aging. They don't usually die of old age, and they have slow metabolisms and reduced oxidative stress. Ming didn't die of old age. It died because someone opened it to count the rings. To count those rings, researchers at Bangor University in Wales had to open the shell. The problem is that opening the shell of a living bivalve is essentially ending its life. At first, the team estimated the animal's age at around 405 years, already an extraordinary number. Only years later, in 2013, did more rigorous analysis using radiocarbon dating and more precise microscopic ring counting revealed the actual 507 years. By then, Ming was already dead. The oldest individually documented animal in history perished not through predation, not through disease, but as a side effect of the very scientific process that was trying to understand it. The irony is almost unbearable. Ming's death was caused by curiosity about its life. Each ring on a quahog's shell represents 1 year of life, formed by the alternation between periods of active growth and periods of seasonal slowdown. In summer, when temperatures rise and food availability increases, the shell grows faster, depositing a wider layer. In winter, growth slows and forms a narrow, dense line. Under a microscope, this alternation creates a pattern of light and dark bands, similar to tree rings but produced by a different mechanism. For a 507-year-old clam, counting those rings requires preparing thin cross-sections of the shell, lighting them properly, and distinguishing each individual band. Painstaking work prone to counting errors that only independent isotopic dating can correct. The species Arctica icelandica, the scientific name of the North Atlantic quahog, is commercially harvested and regularly eaten in soups and chowders throughout New England. Anyone who has had clam chowder in the United States has probably eaten meat from an animal that was several hundred years old, harvested anonymously alongside thousands of others. Most commercially caught quahogs are between 100 and 300 years old. They are sold by the pound, with no reference to their age, and consumed without anyone knowing that the ingredient on their plate was older than most of the nations where it's being served. Ming was not an absolute biological exception. It was simply the oldest specimen found so far. What makes quahog biology so fascinating to aging researchers is precisely the absence of conventional signs of senescence. Senescence is the process by which cells stop dividing correctly, accumulate damage, and begin to function in a degraded way. In humans, this process shows up as visible aging, loss of tissue elasticity, declining organ function, increased vulnerability to disease. In Arctica icelandica, cellular analyses show that very old animals display levels of oxidative DNA damage comparable to those of young specimens. Cells continue replicating faithfully. Metabolism remains stable. The organism simply doesn't age the way most animals do. Oxidative stress is one of the central mechanisms of aging. When cells process oxygen to produce energy, they generate chemical byproducts called free radicals, highly reactive molecules that damage proteins, cell membranes, and DNA itself. The more intense the metabolic activity, the greater the production of free radicals and the faster the accumulation of damage. Organisms with slow metabolisms produce fewer free radicals per unit of time, accumulate damage more slowly, and consequently age more slowly. The quahog combines an extraordinarily low metabolism with efficient antioxidant defense mechanisms, creating an internal biochemistry that seems optimized to minimize the molecular wear and tear that, in other animals, inevitably leads to decline. Ming's death sparked genuine ethical debate within the scientific community. When you capture a rare organism to study it, and the process of studying it kills it, what is the real value of the knowledge gained? The question isn't simple. On one hand, the data obtained from Ming, including climate records preserved in its shell, information about the species' maximum longevity, and tissue samples for molecular analysis, represent a legitimate and lasting scientific contribution. On the other hand, the possibility that Ming could have lived for additional centuries, adding more layers of information to its biological archive, was permanently eliminated. Researchers today are developing non-invasive techniques to estimate the age of bivalves without having to open them. On a geological timescale, 507 years is a blink. On a human scale, however, it represents a historical depth that's hard to wrap your head around. Ming was born before Columbus completed his first voyages to the American continent. When Copernicus published his heliocentric model of the solar system, Ming was 44 years old. When Newton formulated the laws of motion, it was nearly 2 centuries old. When World War II ended, it was 446 years old. Every milestone that organizes human history, wars, discoveries, empires, revolutions, was a contemporary event for Ming, happening while it filtered plankton on the cold floor of the North Atlantic, indifferent to the human noise above. The question scientists still can't answer is, what is the maximum longevity limit for this species? Ming was 507 years old when it was found, but there were no biological signs that it was anywhere near the end of its life. Without knowing whether older specimens still exist on the floor of the Atlantic, and they probably do, it's impossible to establish the species' actual ceiling. Every oceanographic expedition that dredges Icelandic sediments has a theoretical probability of collecting a quahog older than Ming. The question is whether anyone will recognize it before eating it in a soup or destroying it without realizing what it was. Ming may not be the oldest clam ever caught, just the oldest one that someone stopped to examine carefully. Ming's scientific legacy goes far beyond the number 507. Its accidental death directed researchers toward investigating the molecular mechanisms that allow such extreme longevity, driving studies on negligible senescence, low-intensity metabolism, and antioxidant protection in bivalves. Preserved tissue samples continue to be analyzed with techniques that didn't exist in 2006. The shell remains in a scientific repository, available for future analyses using methodologies yet to be developed. An animal that lived 5 centuries in complete silence continues contributing to science even after death. Not because it chose to, but because its extraordinary biology raises questions that science hasn't finished answering. The bowhead whale, Balena mysticetus*, is estimated to live more than 200 years and is possibly the longest-lived mammal. The most concrete proof of this longevity doesn't come from a lab. It comes from a harpoon. In 2007, Inupiat whalers in Alaska found inside the blubber of a bowhead whale 6 iron harpoon tips dated to the late 19th century, manufactured between 1880 and 1895, according to metallurgical analysis. The whale had survived that attack more than a century earlier. A study published in Nature in October 2025 points to an unusually abundant DNA repair protein that allows bowhead whale cells to fix double-strand breaks with speed and precision. In a 200-year-old animal, that precision may be the difference between a healthy body and a tumor. The oldest whale has more than 1,000 times more cells than a human, and almost none of them turn into cancer. Understanding why this is extraordinary requires grasping a central paradox in biology. Large animals should get cancer far more often than small ones. A mouse has billions of cells, a bowhead whale has quadrillions. Every time a cell divides, there's a small probability of a mutation occurring that could eventually lead to tumor growth. Multiply that probability by quadrillions of cells over 2 centuries, and the expected number of cancers would be enormous. Yet bowhead whales rarely develop tumors. Elephants solved a similar problem by accumulating extra copies of the tumor suppressor gene TP53. Naked mole rats have cellular mechanisms that suppress disorganized growth. The bowhead took a different and more efficient path. The protein identified in the study published in Nature in December 2025 is called CIRBP, cold-inducible RNA-binding protein. In bowhead whales, this protein is present at concentrations roughly 100 times higher than in other mammals. To understand what it does, you need to understand the specific type of damage it repairs. Double-strand breaks in DNA. DNA is a double helix molecule, 2 strands wound in a spiral. When both strands rupture simultaneously at the same point, the damage is severe. Human cells repair these breaks, but with an error rate that accumulates over decades. Bowhead whale cells, flooded with CIRBP, repair the same damage with significantly greater speed and precision, accumulating far fewer mutations over the same centuries. The physical evidence of the harpoon is just as eloquent as any molecular data. Iron tips found in the blubber of whales caught in the early 21st century, manufactured with the specific metallurgical technology of the late 19th century, indicate that those animals survived attacks that occurred decades or a century earlier. Iron oxidizes in predictable ways. Analysis of the chemical composition and crystal structure of the metal allows for its manufacture date to be pinpointed with reasonable accuracy. Furthermore, the harpoon models found were specific types that stopped being manufactured after a certain date, making the time window even more precise. The whale's body had encased the metal in successive layers of blubber, isolating it and carrying it as an involuntary record of an attack that was never fatal. Beyond harpoons, there are other methods for estimating the age of bowhead whales. One uses aspartic acid racemization in the eye lens. Amino acids exist in 2 mirror image forms, L and D. Living organisms incorporate almost exclusively the L form. After cell death, this form slowly converts to the D form at a known, temperature-dependent rate. Since the eye lens is not renewed over the animal's lifetime, the ratio between the L and D forms in that specific tissue reveals how long ago those cells were deposited. In other words, the animal's age. Another method analyzes proteins from the baleen, the filtering plates that replace teeth in this species, whose isotopes accumulate in a datable way. Both methods converge on estimates that frequently exceed 200 years. Peto's paradox, the name given to the phenomenon by which large animals don't develop cancer at the expected rate, is one of the most intriguing problems in aging biology. Richard Peto, a British epidemiologist, formalized the observation in the 1970s. If cancer risk were simply proportional to the number of cells, elephants and whales would die of tumors before reaching reproductive maturity. The fact that this doesn't happen suggests that large animals evolved independent tumor suppression mechanisms, each species with its own solution. The bowhead whale represents one of the most elegant solutions. Instead of eliminating damaged cells or multiplying suppressor genes, it simply repairs DNA with such precision that potentially cancerous damage is corrected before it becomes irreversible. The Inupiat people of Alaska, an indigenous people who have practiced subsistence hunting of bowhead whales for millennia, had empirical knowledge of this longevity long before formal science confirmed it. They passed down orally that these animals lived 2 human lifetimes. Knowledge accumulated from generational observations of behaviors, scars, and individually recognizable characteristics over decades. When Inupiat whalers found 19th century harpoons in animals caught in the 21st century, they weren't surprised the way Western scientists were. For them, it was confirmation of something they already knew. Science took decades to arrive at where traditional knowledge had already gotten through completely different means. The most direct implication of the CIRBP research is therapeutic. If a relatively simple protein, present at elevated concentrations, explains a substantial part of the cancer resistance and extreme longevity in bowhead whales, then manipulating the levels of that protein in human cells becomes a legitimate experimental target. The study published in Nature in 2025 demonstrated that introducing bowhead whale CIRBP into human cells improved those cells' ability to repair double-strand breaks in DNA. In Drosophila flies, overexpression of CIRBP extended the insect's lifespan and increased their resistance to radiation. These results don't mean humans will be living 2-century-long lives anytime soon, but they point to a concrete, identified, and testable mechanism linking DNA repair to extreme longevity. The bowhead whale is the second largest animal on Earth, reaching over 80,000 kilograms. It lives exclusively in Arctic and subarctic waters, under the ice or along its edges. It feeds by filtering enormous volumes of water to capture tiny crustaceans, copepods and krill, in quantities sufficient to sustain a body weighing dozens of tons. It migrates seasonally following the ice edge. It has no teeth. It has baleen, horny plates that sieve the water. Its blubber layer can reach 50 centimeters thick, providing thermal insulation in waters near freezing. All of this biology, perfectly adapted to the coldest and most inhospitable environment in the oceans, coexists with a cellular biochemistry that simply refuses to let DNA accumulate damage in any irreversible way. The oldest scientifically estimated specimen of the species reached 211 years, with a margin of error of 35 years, meaning it could have been between 176 and 246 years old when it was caught. That puts the birth of this animal in a window ranging from the mid-18th to the early 19th century, a period when European and American whalers were already hunting this species intensively in the North Atlantic and Arctic. Bowhead whale populations were reduced to a small fraction of their original numbers by commercial hunting. Animals alive in the Arctic today are descendants of survivors from that period, which means the extraordinary longevity of these individuals allowed them to make it through centuries of intense hunting pressure without going completely extinct. The same biology that makes them a scientific curiosity may be part of what kept them alive when extinction would have been the most reasonable expectation. The red sea urchin, Mesocentrodus franciscanus of the North Pacific can live beyond 200 years. And what makes that number extraordinary isn't the figure itself, but what doesn't happen to the organism during that time. The red sea urchin has negligible senescence. It shows no age-related increase in mortality and no decline in reproductive capacity. It grows indefinitely and reproduces throughout its entire adult life. It displays no age-associated telomere shortening and maintains telomerase activity in somatic tissues regardless of age. A 200-year-old sea urchin moves, feeds, and reproduces at rates comparable to those of a young individual. Human aging biology assumes that cells accumulate damage over time, telomeres shorten, and functions decline. The red sea urchin operates by a different logic, one that gerontologists still haven't been able to fully replicate or explain. To understand what negligible senescence means in practice, it helps to compare it with what happens in organisms that age the conventional way. In humans, somatic cells, body cells that aren't reproductive, divide a limited number of times before entering a state of senescence or dying. This limit exists because with each division, telomeres, the protective structures at the ends of chromosomes shorten slightly. When they get too short, the cell stops dividing. This mechanism protects against uncontrolled proliferation, cancer, but comes at a cost. The organism progressively loses its ability to regenerate tissue. The red sea urchin seems to bypass this trade-off entirely, keeping telomeres stable over centuries through continuous telomerase activity in somatic tissues. Telomerase is an enzyme that rebuilds telomeres after each cell division, compensating for the wear. In humans, this enzyme is highly active in germ cells, the cells that produce eggs and sperm, but is virtually absent in most adult somatic tissues. In young organisms, some residual telomerase activity contributes to growth and repair, but it diminishes over time. In the red sea urchin, analyses of multiple adult specimens indicate that telomerase activity is maintained in somatic tissues regardless of the animal's age. It's not just that the sea urchin doesn't age, it's that its cellular machinery seems to actively prevent the shortening that would trigger aging. The difference between aging and not aging, in this case, comes down to a single enzyme that keeps working when it should have stopped. Indeterminate growth is a direct consequence of this biology. Most vertebrate animals, and many invertebrates, reach a genetically defined maximum size, after which growth stops. This ceiling stems in part from cellular senescence. When cells stop dividing efficiently, tissues stop expanding. The red sea urchin has no such ceiling. Older specimens are typically larger than young ones, and the correlation between size and age is robust enough to be used as an age estimation tool. The larger the animal, the older it probably is. This doesn't mean they grow indefinitely in practice. Environmental constraints, food availability, and predation mortality set real limits. But biologically, nothing in the organism tells it to stop growing. Lifelong reproduction represents another radical departure from familiar patterns. Female mammals are born with a fixed number of ovarian follicles that are used up over the course of their reproductive lives. When the supply runs out, menopause begins and fertility ends. This process is directly tied to reproductive aging. In the red sea urchin, gonads continue producing viable gametes in very old specimens. Studies that measured gamete production and quality across different age groups found no age-correlated decline. A 200-year-old animal produces functional gametes with efficiency comparable to that of a 20-year-old. This reproductive continuity implies that the cellular maintenance systems supporting the gonads work equally well throughout that entire period. The sea urchin's nervous system behaves differently from its other tissues, and that's significant. Research analyzing gene expression profiles in the muscle and esophagus of old and young animals found very few differences. Stable genetic transcription is considered one of the molecular hallmarks of negligible senescence. However, in the radial nerve, the sea urchin's central nervous structure, more than 3,000 genes with significantly altered expression were identified with age, including genes involved in synaptogenesis, axonogenesis, and neuroprotection. Rather than degradation, the nervous system appears to actively activate neuronal preservation processes over time. The organism doesn't just maintain function, it seems to actively compensate for wear before it becomes noticeable. Comparing sea urchin species with radically different lifespans provides clues about the mechanisms involved. The green sea urchin, Lytechinus variegatus *, a closely related species, has a maximum lifespan of just 4 years. The longevity gap, 4 years versus more than 200, between species belonging to the same taxonomic group suggests that relatively focused genomic changes can produce dramatic effects on lifespan. Comparative genome analyses of long-lived and short-lived species identified expanded gene families in the long-lived ones. Genes related to innate immunity, the sensory nervous system, and genomic stability are present in more copies, increasing defensive and repair capacity over centuries. There's an ecological irony in this extraordinary longevity. The red sea urchin is eaten as a delicacy in various cultures. Its gonads, known as uni in Japanese cuisine, are one of the most prized ingredients in upscale restaurants around the world. Commercially caught specimens are rarely analyzed for age before consumption. It's unlikely but not impossible that organisms decades or even centuries old are collected and eaten without anyone noticing. Extraordinary longevity doesn't give the animal any visible status that would distinguish it from a young one on the shelf of a seafood market. For gerontologists, scientists who study aging, the red sea urchin represents a productive puzzle, an animal organism that shares fundamental cellular mechanisms with humans, including telomeres and telomerase, but uses those mechanisms in a radically different way. Pinpointing exactly which genetic and molecular differences allow telomerase to remain active in somatic tissues for centuries without triggering unchecked tumor proliferation could shed light on why telomerase was evolutionarily suppressed in humans and what the costs and benefits of therapeutically reactivating it might be. The animal that doesn't age functions as a negative map of human aging. By showing what is biologically possible, it defines with greater precision what still needs to be explained. The red sea urchin inhabits rocky shores of the North Pacific, from Baja California to Alaska, at depths ranging from the intertidal zone to more than 90 meters below the surface. It feeds primarily on algae, moving slowly on its spines and tube feet, hydraulic structures that function like microscopic suction cups. Its existence is quiet, slow, and deeply compatible with longevity. The moderate temperatures of a temperate ocean, a consistent diet, and the absence of a high metabolic rate. The paradox it embodies, being a simple animal that solves one of biology's most complex problems, doesn't shrink with knowledge. It deepens. The more one understands about the molecular biology of aging, the more impressive the solution this spiny echinoderm found long ago, alone at the bottom of the Pacific, turns out to be. Turritopsis dohrnii, the immortal jellyfish, measures 4.5 millimeters in diameter and represents the only known case of an animal capable of completely reversing its life cycle after reaching sexual maturity. When Turritopsis dohrnii suffers physical damage or faces starvation, it can survive through a unique process called transdifferentiation. It undergoes a kind of rebirth and transforms back to the beginning of its life cycle as a polyp. During transdifferentiation, the medusa's cells reprogram themselves to become polyp cells, essentially erasing the jellyfish's adult form and starting over from scratch. The species possesses unique mechanisms related to telomere maintenance, which play a significant role in its regenerative capabilities. Dohrnii maintains telomere length through specific cellular processes during life cycle reversal, effectively resetting cellular aging. The process has never been observed in the wild. It happens too fast, and the observation windows are practically impossible to catch. The typical jellyfish life cycle includes two main forms, the polyp and the medusa. The polyp is the stage attached to a substrate. It looks like a small anemone, motionless, fixed to rocks or surfaces. The medusa is the free-swimming stage, what most people picture when they think of a jellyfish. A gelatinous body with tentacles pulsing through the water column. In most cnidarian species, the cycle is one-directional. Polyps produce medusae by budding, medusae reproduce sexually, and the resulting larvae settle and form new polyps. The death of the medusa ends that individual cycle. Turritopsis dohrnii breaks that one-directionality. The adult medusa, under stress, can revert to the polyp stage, restarting the entire cycle without dying. The cellular mechanism by which this occurs is called transdifferentiation, and it's extraordinary even outside the context of longevity. Transdifferentiation is the process by which an already specialized cell, committed to a specific function, transforms into a completely different cell type, without passing through the intermediate stage of an undifferentiated stem cell. In the lab, inducing transdifferentiation in mammalian cells requires elaborate molecular interventions and still results in extremely low efficiency. Turritopsis dohrnii carries out this process at a whole-body scale, spontaneously, transforming the medusa's muscle cells into epithelial cells of the polyp, nerve cells into other types, completely reconfiguring its cellular identity within 24 to 72 hours. During the transition, the animal passes through an intermediate state described as a cyst, a compact cellular mass with no visible characteristics of either a medusa or a polyp. Gene expression analyses at this cyst stage revealed distinct patterns. Genes related to DNA repair, telomere maintenance, and chromatin remodeling are significantly more active in this phase than in either the polyp or the mature medusa phases. Chromatin is the complex of DNA and proteins that forms the chromosomes. Its remodeling is the mechanism by which cells change which genes are being actively read. Remodeling chromatin at a body-wide scale is the equivalent of rewriting the operating program of every cell in the organism. That's what allows the cyst to become a polyp instead of simply dying. The word immortal requires precise qualification. Turritopsis dohrnii is not immortal in the sense that it cannot die. Predators eat it. Disease can kill it. Unfavorable environmental conditions can be lethal before the reversal process is completed. The immortality in question is biological and theoretical. In principle, there is no intrinsic limit to the number of times the cycle can be reversed. The organism doesn't carry an internal countdown, doesn't accumulate molecular aging markers that become irreversible. Each time it completes the reversal and becomes a polyp again, it starts the cycle from zero. Cells with restored telomeres, rejuvenated gene expression patterns, with no biochemical memory of previous cycles. What limits its life in the wild is not aging, but the environment. The discovery of Turritopsis dohrnii's life cycle reversal capability was published in 1996 by Ferdinando Boero and collaborators, but the phenomenon had been observed informally decades earlier without its implications being recognized. The species was first described in 1883 in the Mediterranean Sea. For over 100 years, it coexisted with marine biologists without anyone noticing what it was capable of, perhaps because the scale of the phenomenon, combined with the animal's tiny size and the speed of the process, made accidental observation extremely unlikely. Today, the species is known to have a global distribution, transported in ships' ballast water to oceans where it didn't originally exist, with established populations in the Mediterranean, Atlantic, and Pacific. The life cycle reversal has never been documented in the wild, only in the lab under controlled conditions. This doesn't mean it doesn't happen in the ocean. It means it's practically impossible to observe. A 4.5 millimeter jellyfish running into trouble in the middle of the open ocean and beginning a process of collapse into a microscopic cyst is not something researchers can detect without being looking directly at the animal at exactly the right moment. In the lab, the reversal can be deliberately induced by physical stress, piercing the medusa with a needle, or by food restriction. The process completes itself in under 3 days, resulting in a polyp genetically identical to the original medusa, ready to restart the cycle. The implications for regenerative medicine are the subject of active research, but the obstacles are considerable. What Turritopsis dohrnii does naturally, reprogramming differentiated cells into new states, is exactly what researchers are trying to achieve artificially to treat degenerative diseases, regenerate damaged tissues, or reverse aspects of aging in human cells. The difference is that the animal does this throughout its entire body simultaneously, with a level of coordination that appears to be genetically encoded in ways that haven't yet been fully mapped. Identifying the gene networks that control this process and translating them into therapeutic contexts in mammals remains a scientific challenge of enormous magnitude. Fascinating precisely because the model exists, works, and fits on the tip of a finger. Turritopsis dohrnii belongs to the group of hydrozoans, cnidarians with gelatinous bodies whose anatomical simplicity contrasts sharply with the sophistication of the process they carry out. It has no brain, only a diffuse nerve net distributed throughout the body. It has no blood and no organs in the conventional sense. Its body is composed essentially of two cell layers separated by a gelatinous substance called mesoglea. Within this architecture of extreme simplicity, the molecular mechanisms of transdifferentiation operate with remarkable precision. Neurological complexity is not a prerequisite for extraordinary biological capabilities, a lesson that T. dohrnii illustrates more clearly than any other known animal. The central paradox of Turritopsis dohrnii is that its immortality mechanism is activated precisely when the animal is under existential threat. It is not a strategy for expansion or dominance. It is a last resort survival strategy, triggered when all other options are exhausted. Healthy, well-fed animals with no physical damage don't reverse the cycle. They complete it normally, reproduce, and eventually die as aging medusae. The reversal is the response to imminent failure, not to success. This suggests that in the wild, most individuals of the species never actually activate the mechanism that could make them potentially immortal, and they die without ever using it. The tool exists. It just doesn't always get used. The Greenland shark has an estimated lifespan of up to 400 years. It's found in the cold waters of the Arctic and North Atlantic and is known for its slow growth rate, taking up to 150 years to reach sexual maturity. To put that in perspective, a Greenland shark that reached sexual maturity at 150 years was born before the French Revolution. Researchers date its age using the eye lens, which accumulates inert tissue from birth. The older the animal, the more ancient the protein at the center of the lens. The oldest specimen dated was estimated at 392 years, with a margin of error of 120 years in either direction, which puts the upper limit at 512 years. It frequently goes blind. Parasites, *Omidocoida elongata*, attach to the cornea and cause permanent vision damage. Extreme longevity doesn't require speed or sharp eyesight. It requires a slow metabolism, low temperatures, and the absence of predators. Dating the age of a fish normally uses calcified structures, vertebrae or otoliths, inner ear bones, which deposit annual layers analogous to tree rings. The Greenland shark has neither calcified vertebrae nor otoliths suitable for this technique. To get around the problem, Julius Nielsen and collaborators published a method in 2016 in the journal Science using an alternative approach: radiocarbon dating of proteins from the eye lens. The center of the lens is formed during embryonic development and is never replaced over the animal's lifetime. It is metabolically inert tissue from birth. Measuring the proportion of carbon-14 in this tissue makes it possible to determine when the animal was born, using as a reference point the pulses of carbon-14 released by atmospheric nuclear tests in the 1950s, which left a datable isotopic signature in the ocean. The Greenland shark's cruising speed is approximately 0.3 meters per second, about 1 kilometer per hour, a pace comparable to a person walking very slowly down a hallway. No other fish of similar size moves so slowly on a sustained basis. When corrected for body size, this speed is the lowest recorded among all known fish. This is not an accidental limitation. It's a direct consequence of the ambient temperature. Water near freezing slows biochemical reactions, reduces metabolism, and diminishes the capacity for muscle contraction. The shark adapts to this environment not by fighting it, but by becoming as metabolically slow as the water around it. The energy cost of each movement is minimal. The need for food is correspondingly low. Greenland shark meat is toxic when fresh. It contains high concentrations of trimethylamine oxide, TMAO, and urea, compounds that function as a biochemical antifreeze, stabilizing proteins under pressure and temperatures near zero. When ingested by humans, these compounds cause symptoms similar to alcohol intoxication, followed by more serious effects. In Iceland, there is a traditional dish called hakarl, made from Greenland shark meat that is fermented and buried for months to break down the toxins before eating. The same compounds that make the meat toxic are part of the system that allows the animal to exist in waters that would kill other predators. The toxicity is not a byproduct, it's an adaptive solution. The relationship with the parasite Ammodocoida elongata is one of the strangest biological associations known. This copepod, a tiny crustacean, a distant relative of crabs and lobsters, attaches itself to the shark's cornea, feeding on corneal tissue and causing ulceration, mineralization, and swelling. Most Greenland sharks in the North Atlantic have at least one of these parasites in their eyes. Many have parasites in both. The females of the copepod, which are larger, are the ones that attach permanently. They can reach a few centimeters in length and are visible in photographs as appendages dangling from the shark's eye. One still unconfirmed hypothesis suggests that the parasite's bioluminescence could attract prey toward the nearly blind shark, a parasite-host relationship that would be, paradoxically, mutually beneficial. Despite impaired vision, the Greenland shark is an effective predator. Stomach contents of captured specimens have revealed whole polar bears, reindeer, moose, and seals, in addition to fish and cephalopods. The ability to capture large, agile prey without seeing properly is explained by its primary reliance on smell and hearing, senses that work exceptionally well in the dark, cold Arctic Ocean. Shark olfaction detects traces of blood or organic compounds at vanishingly small concentrations over long distances. Lateral lines, sensory organs that detect pressure variations in the water, further compensate for the lack of vision. Research published in 2026 in Nature Communications revealed that, despite the corneal parasites, the shark's retina remains intact and adapted to low light. Vision is obscured by the damaged cornea, not destroyed at the source. Dating the oldest specimen produced an estimate of 392 years, with a 95% confidence interval between 272 and 512 years. Even at the lower end of that range, the animal was born before Isaac Newton published the Principia Mathematica. At the upper end, it was born before Galileo Galilei built his first telescope. The 120-year uncertainty, striking for a dating technique, reflects variations in carbon-14 concentrations across the Arctic Ocean and uncertainties about the rate of protein deposition in the embryonic lens. Despite the margin, the 2016 study analyzed 28 individuals and found consistent patterns that make the species' extreme longevity scientifically incontestable, even if the exact age of each individual carries considerable uncertainty. The Greenland shark's genome, published in 2024, revealed molecular characteristics that are beginning to explain its longevity. Among the most unusual findings is a massive expansion of genes encoding ferritin, the protein that stores iron in cells. The genome contains more than 150 copies of the light chain ferritin gene and nearly 180 copies of the heavy chain, organized into large clusters. In humans, only a handful of copies of these genes exist. Excess ferritin may contribute to protection against oxidative stress, one of the most studied pathways of aging. Excess free iron in cells produces highly damaging free radicals. Sequestering that iron in ferritin reduces the damage. The genome also shows alterations in genes related to DNA repair and tumor suppression, suggesting molecular solutions that converge with those found in the bowhead whale. There's something conceptually unsettling about the Greenland shark's longevity that goes beyond the numbers. This animal was born, possibly, before any person alive today existed. It spent centuries in the dark depths of the Arctic while empires rose and fell above it. Wars redrew borders, and technologies transformed civilizations. None of that reached the ocean floor. The shark kept moving at 1 kilometer per hour, blind, in near-freezing temperatures, unaware of and unbothered by the noise above. Its existence is not dramatic. It is the opposite of drama. It is silent, metabolic persistence, without urgency or purpose beyond continuing. And it continued for centuries doing exactly that. What connects the Greenland shark to the other long-lived animals in this series? The glass sponge, the Ming clam, the bowhead whale, the sea urchin, the jellyfish, is a convergence of strategies that aging biology is only beginning to map with molecular precision. Slow metabolism, low temperatures, efficient DNA repair, telomere maintenance, reinforced antioxidant protection, the absence of premature cellular senescence. Each species arrived at a different combination of these elements through distinct evolutionary paths, but all of them point to the same conclusion. Aging is not a universal physical inevitability. It is a specific biological solution to a specific problem, and different solutions produce radically different lifespans. What the oceans conceal in their cold, dark depths are organisms that found other answers to the same question that human medicine is still learning to formulate with precision. The golden coral, Gerardia sp*, is one of the rarest and most impressive deep-sea corals, and one of the longest-lived animals ever precisely dated. Colonies found in the deep waters around Hawaii were radiocarbon dated at approximately 2,742 years old, older than the Roman Empire. Unlike shallow water corals, which depend on symbiotic algae and sunlight, the golden coral filters particles directly from the water, which allows it to live at depths far beyond the reach of light. Its growth rate is extraordinarily slow. A single colony may grow only a few centimeters per century. This has historically made them a target for collection as deep-sea jewelry, a practice now banned in most jurisdictions. A colony that grew a few centimeters while entire empires rose and collapsed above it. To understand what makes that number so remarkable, it helps to understand what a coral actually is. The word coral refers both to the individual animal, called a polyp, and to the collective structure these animals build. Each polyp is a tiny organism with tentacles that secretes material around itself to form the colony's skeleton. In shallow water corals, that skeleton is made of calcium carbonate, a mineral similar to marble. In the golden coral and other deep-sea corals with protein-based skeletons, the structural material is a tough protein called gorgonin. This protein incorporates carbon from the surrounding water during deposition, which allows for radiocarbon dating with considerable precision. The colony grows centimeter by centimeter, layer by layer, each added ring recording the chemical composition of the water at that moment. The dating that revealed 2,742 years was published in 2009 by Brendan Rourke and collaborators at Texas A&M University. The study analyzed Hawaiian specimens of Gerardia sp. and the black coral Leiopathes sp., finding even greater ages for the latter, approximately 4,265 years. Previous estimates, based on counting what appeared to be annual rings in the skeleton, had pointed to a maximum of 70 years for the golden coral, a figure researchers had assumed without independent verification. Radiocarbon dating revealed that these rings were not annual. They represented much longer periods. The error was not a small one. The difference between 70 years and 2,742 years is a factor of nearly 40. one of the largest gaps between estimate and reality ever documented in marine biology. The radial growth rate measured for Hawaiian golden coral falls between 14 and 45 micrometers per year, roughly 0.015 to 0.045 millimeters annually. To make that scale tangible, a human hair is between 60 and 80 micrometers in diameter. The golden coral grows, each year, a fraction of the width of a single strand of hair. Over a century, it accumulates somewhere between 1 and 4 millimeters of radial thickness. Over 1,000 years, perhaps 1 to 4 centimeters. The visible structure of a 2,000-year-old colony therefore represents only a few centimeters of accumulated growth, which explains both its delicate appearance and the ease with which it can be irreversibly destroyed by a single pass of a deep-sea trawling net across the ocean floor. Deep-sea corals like the golden coral inhabit underwater mountains, seamounts, and continental margins at depths between 300 and 3,000 meters. Without sunlight, they can't host photosynthetic algae the way tropical reef corals do. They feed on organic particles that sink from the ocean surface, fragments of dead organisms, zooplankton fecal matter, decomposed material. This particle flow, known as marine snow, is slow and irregular. Food availability in the abyss is a tiny fraction of what exists in shallow waters. A correspondingly low metabolism allows them to survive on this sparse supply, and it's precisely that reduced metabolism that, as with the other organisms in this series, contributes to extraordinary longevity. The commercial collection of golden coral for jewelry manufacturing represented a serious and nearly invisible threat to these colonies. For decades, ships equipped with dredges and nets harvested precious corals from seamounts in the Pacific and Atlantic, selling the material to craftspeople who produced bracelets, necklaces, and sculptures. A colony destroyed by collection represents millennia of growth eliminated in seconds. No restoration effort would be capable of recreating what was removed on any human timescale. The colony simply would not grow back within any human lifetime. Today, international regulations prohibit collection in many areas, but enforcement in deep waters remains technically difficult and financially costly. Beyond direct collection, deep-sea bottom trawling represents an even more widespread threat. Nets dragged across seamounts indiscriminately destroy all sessile fauna, organisms fixed to the substrate, including centuries and millennia-old corals with no immediate commercial value. A single trawl pass can eliminate entire communities that took centuries to establish. In many cases, the corals destroyed were unknown to science, never sampled, identified, or studied. Their local extinction occurred before their existence was even documented. Deep-sea corals are, in this sense, doubly vulnerable. They grow far too slowly to recover, and they live in environments that are rarely observed before they are disturbed. The deep-sea environment where the golden coral persists is remarkably stable. Temperature stays near 4 degrees Celsius with minimal variation over centuries. Pressure remains constant. The absence of sunlight eliminates the circadian variations that affect surface organisms. Currents are slow and relatively predictable. This set of stable conditions is analogous to the environments of the other long-lived species discussed in this series: ocean abyss, Arctic waters, Pacific seafloor. Environmental stability appears to be a fundamental permissive condition for extreme longevity. Less external variation means less demand for internal adaptation, less physiological stress, and less accumulated damage. The combination of extreme longevity, ultra-slow growth, and sensitivity to disturbance positions the golden coral as an unwitting sentinel of ocean conditions over millennia. Isotopic analyses of the protein skeletons of these colonies can, in principle, reconstruct variations in temperature, productivity, and chemical composition of the ocean with temporal resolution not available from other natural archives at that depth. Each layer of gorgonin deposited preserves a chemical signature of the environment during that specific period, a kind of chemical diary written in protein, stretching back more than 2 millennia. Destroying a golden coral colony is not just eliminating an ancient organism, it's erasing an irreplaceable oceanographic archive. The golden coral colony, dated at 2,742 years in Hawaii, was born around the 8th century BCE, a period when Greek philosophers were beginning to formalize the first theories about the nature of the universe. Since then, it has grown a few centimeters while the world above it transformed completely. Empires rose and fell, languages were born and died, continents were mapped, atoms were discovered. The colony never knew any of it. It filtered particles, deposited protein, grew imperceptibly. At the dark bottom of the Pacific, time operates on a different scale, and the golden coral inhabits that scale with complete indifference to what happens above, near the surface where human life passes by in a flash. Analysis of the bowhead whale's genome identifies genes under positive selection and species-specific mutations in genes linked to cancer and aging, as well as gains and losses of genes associated with DNA repair, cell cycle regulation, cancer, and aging. Peto's paradox states that larger animals, with more cells and more cell divisions over a longer life, should develop more cancer. The bowhead whale weighs 80,000 kilograms and lives 200 years. It should be a tumor-producing machine. Large whales, with more than 1,000 times more cells than humans, don't appear to have an increased cancer risk, suggesting the existence of natural mechanisms capable of suppressing cancer more effectively than those of other animals. Understanding these mechanisms is one of the most active lines of research in oncology today. Peto's paradox has a name because it contradicts the most intuitive expectation in oncology. Cancer begins when a cell accumulates enough mutations to escape the normal controls on growth and division. The more cells an organism has and the longer it lives, the more cell divisions occur, and the greater the cumulative probability that some cell will accumulate the critical set of mutations. A mouse with billions of cells and a 2-year lifespan should have a much lower cancer risk than a human, with trillions of cells and an 80-year life. And a human should have an astronomically lower risk than a bowhead whale, with quadrillions of cells and a 2-century lifespan. The data, however, don't confirm this progression. The cancer mortality rate in whales doesn't appear proportional to what the arithmetic of DNA would predict. The study published in Nature in December 2025 produced an initially counterintuitive result. Researchers at the University of Rochester induced oncogenic transformation in fibroblasts, connective tissue cells, from different species: mice, cows, humans, and bowhead whales. Whale fibroblasts required fewer oncogenic interventions to transform into cancer cells than human fibroblasts. This suggests that, paradoxically, whale cells are more susceptible to initial transformation, not more resistant. What the whale has in abundance is not resistance to cancer initiation, but an exceptional ability to repair DNA before mutations accumulate to the point of triggering that transformation. The distinction between preventing the onset of cancer and repairing damage before it begins is fundamental. In humans and elephants, one strategy is to keep tumor suppressor genes highly active. Genes like TP53 that identify damaged cells and eliminate them through apoptosis program cell death. Elephants have multiple copies of TP53. Humans have one. This strategy eliminates problematic cells before they become tumors. The bowhead whale took a different path. Instead of eliminating damaged cells, it repairs the damage with such precision that cells rarely need to be eliminated at all. TP53 activity in whale cells is actually lower than in humans, but that doesn't matter because the DNA arrives far less damaged at the point where TP53 would need to act. The protein CIRBP cold-inducible RNA-binding protein, is present at concentrations roughly 100 times higher in bowhead whale cells compared to other mammals. Its name gives away its origin. It's a protein originally identified for being activated by cold. In land mammals, CGRBP responds to temperature drops as a transient cellular protection mechanism. In the bowhead whale, which permanently inhabits waters near freezing, this protein is constitutively expressed at very high levels. The constant cold of the Arctic environment may have selected, over generations, for lineages with increasingly elevated CIRBP expression, and that elevation brought with it better DNA repair and lower mutation accumulation as a consequence. The experiments in the 2025 study directly tested whether whale CIRBP improves DNA repair in cells of other species. When researchers introduced the bovine version of CIRBP into human cells, the repair of double-strand breaks improved. When they introduced the bowhead whale-specific version, which differs in sequence from the human version, the effect was even more pronounced. In Drosophila melanogaster flies, overexpression of CIRBP extended the insect's lifespan and increased their resistance to ionizing radiation, which damages DNA in a manner analogous to the accumulation of somatic mutations. The protein doesn't just work in the original species. It works when transferred to radically different organisms, suggesting that the underlying mechanism is fundamental enough to operate across distinct biological contexts. Oncology has been mapping Peto's paradox for decades precisely because it offers a potentially powerful clue. If nature has solved the problem of suppressing cancer in organisms far larger and longer lived than humans, the mechanisms used may be therapeutically adaptable. Identifying those mechanisms in whales, elephants, and naked mole rats, other organisms with remarkable cancer resistance, is part of a scientific strategy of learning from solutions that biology has already tested at an evolutionary scale. Each long-lived species with a low cancer incidence is, in this sense, a natural experiment that lasted millions of years and may contain answers more sophisticated than any laboratory experiment could produce in decades. The fact that bowhead whale cells display lower mutation rates than human cells under the same conditions is, in itself, an extraordinary result. Mutations are the fuel of cancer. Without the accumulation of mutations in critical genes, the cascade that leads to malignant transformation never completes. If whale cells accumulate mutations more slowly, the cancer risk doesn't decrease linearly with the number of mutations. It decreases far more sharply, because many of the critical mutation combinations simply never occur over 2 centuries of life. The precision of DNA repair is not a minor quantitative difference. It's a difference that can determine whether an animal with quadrillions of cells lives 2 healthy centuries or succumbs to tumors decades earlier. There is a rarely discussed ecological dimension to this biology. The bowhead whale lives exclusively in Arctic and subarctic waters, one of the coldest environments on the planet, and one exposed to intense seasonal ultraviolet radiation, especially during the polar summer when the sun doesn't set for weeks. UV radiation damages DNA. Marine animals at the surface are regularly exposed to this damage. The combination of high seasonal UV exposure with superior capacity to repair DNA damage may have been an important selective pressure. Lineages with better CIRBP survived and reproduced more efficiently in an environment where DNA damage was constant and unavoidable. The Arctic, paradoxically, may have forged the very mechanism that made the bowhead whale the longest-lived known mammal. Researchers who study the bowhead whale often note that each individual captured for analysis represents a unique and unrepeatable scientific opportunity. Tissue samples from these whales are rare. The species is internationally protected, and only indigenous communities such as the Inupiat in Alaska are permitted to carry out traditional subsistence hunting. Each legally obtained sample secured with community consent, fuels years of research across multiple labs simultaneously. The genome, sequenced and analyzed, revealed not just CIRBP, but a complete map of molecular adaptations. Changes in cold response genes, cell cycle regulation, lipid processing, and DNA repair that, taken together, paint the picture of an organism optimized to persist in a hostile environment for centuries without its own DNA betraying it. The red sea urchin, the quahog clam, the jellyfish Turritopsis dohrnii, and certain sponges share a characteristic that gerontologists call negligible senescence, the absence of measurable functional decline over time. Both long-lived and short-lived sea urchin species show no age-associated telomere shortening and maintain telomerase activity in somatic tissues regardless of age. Research into the molecular biology of long-lived sponges, such as Monoraphis chuni, suggests that these organisms maintain high levels of telomerase activity and efficient DNA repair mechanisms. This allows them to avoid the cellular damage and telomere shortening that typically lead to programmed cell death in other species. Human aging is treated by medicine as inevitable. For these marine organisms, it seems to be optional. Negligible senescence is not immortality. It is the absence of measurable functional decline attributable to age. Organisms with negligible senescence still die through predation, infection, accident, environmental disturbance. What doesn't happen is the progressive decline of bodily functions that, in organisms with conventional senescence, makes the organism progressively more vulnerable over time. In humans, this decline manifests as reduced muscle strength, cognitive decline, a weakening immune system, increased bone fragility, all processes that accelerate mortality in older age groups. Organisms with negligible senescence don't follow these trajectories. The probability of dying in any given year doesn't increase as the animal ages. It remains roughly constant, driven by external factors, not internal ones. The most consistently identified common denominator among long-lived marine organisms is the maintenance of telomeres in somatic tissues throughout life. In humans and most mammals, telomeres shorten with each cell division in somatic tissues because telomerase, the enzyme that rebuilds telomeres, is suppressed in those tissues after initial development. This suppression appears to have been selected as protection against cancer. Cells that divide indefinitely without a telomere limit are more likely to accumulate enough mutations for malignant transformation. The cost of that protection is aging. Cells that stop dividing efficiently produce tissues that regenerate increasingly poorly. Organisms with negligible senescence appear to have found ways to keep telomerase active without incurring a proportional cancer risk. Research into why organisms with negligible senescence don't develop cancer at elevated rates, despite maintaining long telomeres and cells with high replicative capacity, is one of the most productive problems in gerontology today. Some hypotheses under investigation include alternative tumor suppression mechanisms that don't rely on telomere shortening as an alarm signal, greater DNA repair efficiency that reduces the rate of mutation accumulation independently of cell division, and more efficient immune systems in detecting and eliminating precancerous cells before they proliferate. Different species likely combine these mechanisms in different proportions, which is why very different organisms converge on negligible senescence without using exactly the same molecular solution. The list of organisms recognized as having negligible senescence is longer than most people realize. Beyond the marine animals in this series, it includes the Aldabra giant tortoise, the Tuatara lizard of New Zealand, some species of ornamental carp, and the Mekong giant catfish. What these organisms have in common, beyond the marine ones, is frequently some combination of slow metabolism, low or constant ambient temperature, the absence of significant natural predators, and longevity that predisposes toward the selection of more robust cellular maintenance mechanisms. Negligible senescence is not exclusive to aquatic environments. It is, however, especially common in them, possibly because cold water, high pressure, and food scarcity create selective pressures that favor organisms capable of persisting for centuries rather than growing, reproducing, and dying quickly. There is a fundamental evolutionary tension behind senescence. The disposable soma theory, proposed by biologist Thomas Kirkwood, argues that organisms allocate limited resources between reproduction and bodily maintenance. Reproducing early and often, before predators, disease, or accidents eliminate the individual, is an advantageous strategy in environments with high extrinsic mortality. Investing heavily in bodily maintenance for longevity only pays off if the probability of surviving to advanced ages is high enough. Organisms in stable environments, with few predators and relatively reliable resources, can afford the evolutionary luxury of investing in long-term maintenance, and that selection produces negligible senescence over enough generations. Turritopsis dohrnii adds an additional dimension to the concept. While sea urchins, sponges, and clams appear to simply slow aging to imperceptible rates, the immortal jellyfish seems to actively reverse it. During the transition to the cis stage, genes related to telomere maintenance and DNA repair are significantly more expressed than in adult phases. The life cycle reversal is not just a deceleration of aging, it is a molecular reset with restored telomeres and rejuvenated gene expression patterns. If negligible senescence is a car that never loses speed, dohrnii is a car that, when it slows down too much, simply restarts the engine and begins again from the starting line. The most direct implication for human aging biology is not that humans should live for millennia. It's that aging, as a biological process, is far more malleable than traditional clinical medicine had assumed. For decades, gerontology treated aging as inevitable degeneration, focusing on managing symptoms and slowing complications. The discovery of negligible senescence in multiple independent lineages, from sponges to whales, passing through sea urchins and clams, demonstrates that animal organisms can, in principle, maintain full biological function for periods far greater than what was considered biologically possible until recently. This doesn't guarantee that interventions in humans are feasible or safe, but it establishes that the problem has a biological solution, because nature has already found it multiple times. The organisms with negligible senescence that inhabit deep oceans share more than molecular mechanisms. They share a radically different existential logic from that of humans. Humans face intense time pressure, limited reproductive windows, businesses that rise and fall in decades, cultures that transform within generations. Human biological time is marked by urgency. Sponges, corals, and abyssal sea urchins inhabit a time where centuries are natural units of measure, where a single generation may outlast any recorded human civilization. It's not that these organisms are superior, it's that they inhabit a completely different timescale, and their molecular solutions for persistence are calibrated to that scale, not ours. Studying negligible senescence in marine organisms requires methodologies that biology is still developing. Dating specimens with precision is the first obstacle. Without reliable age markers, comparisons between young and old individuals become impossible. Techniques such as radiocarbon dating, amino acid racemization, and isotopic analysis of accumulative structures like spicules, shells, and protein skeletons have advanced significantly in recent decades. The second obstacle is accessing these organisms. Living at hundreds or thousands of meters of depth makes collection and observation expensive and technically demanding. The third is temporal. Longitudinal experiments on organisms that live centuries are impractical for any single generation of researchers. The science of marine longevity depends on intergenerational collaboration. Data collected by a researcher today will be analyzed, perhaps, by a researcher not yet born. Monoraphis chuni, at 11,000 years old, was sitting at the bottom of the Indo-Pacific when the first farmers in the Middle East were planting the first crops. The Hawaiian golden coral grew a few centimeters while entire civilizations rose and disappeared. The Ming clam was born while Leonardo da Vinci was painting and died in the lab that was trying to understand it. The bowhead whale carried 19th century harpoons in its body for over 100 years. The 200-year-old red sea urchin reproduces at the same rate as a young one, and Turritopsis dohrnii, at 4.5 millimeters, can restart its own life cycle indefinitely, as long as it isn't eaten first. The ocean is home to some of the longest-lived creatures on Earth, and the common denominator is stability. Constant temperature, absence of predators, slow metabolism, and environments that don't change for millennia. The formula for extreme longevity in the ocean is simply, do less more slowly for longer. The narrative these 6 organisms build together is not about curious biological exceptions. It's about what life can be when it isn't under the pressure of urgency. Humans and most land mammals face pressures that select for rapid growth, early reproduction, and the ability to respond to immediate threats. That set of pressures produces agile, adaptive, relatively short-lived organisms. The deep ocean operates under the opposite logic. Stability instead of variation, scarcity instead of abundance, absence of predators instead of constant predation. These conditions select for organisms that invest in long-term maintenance rather than rapid growth. And the result is creatures that exist on timescales that make any human notion of longevity seem momentary. Slow metabolism appears as the through line in all 6 cases. The glass sponge filters microparticles at a rate that consumes minimal energy. The golden coral grows micrometers per year. The Ming clam processed oxygen at rates far lower than those of smaller, short-lived bivalves. The bowhead whale, despite its size, has a specific metabolic rate per kilogram of tissue that is extraordinarily low for a mammal of its size. The red sea urchin exhibits low levels of cell proliferation and apoptosis in internal tissues, consistent with a reduced metabolism. The Greenland shark moves at 1 kilometer per hour in near-freezing water. Low metabolism reduces the production of free radicals, reduces the rate of cell division, reduces the accumulation of mutations, and with each of those reductions, expected longevity increases. Low temperature amplifies this effect. Chemical reactions, including biochemical reactions inside cells, occur more slowly at lower temperatures. Water at 4 degrees Celsius slows the metabolism of any organism that doesn't actively regulate its internal temperature, which includes all the marine invertebrates in this series and the Greenland shark. Even the bowhead whale, a warm-blooded mammal that maintains a relatively stable body temperature, inhabits an environment so cold that the selective pressure for efficient cellular maintenance is amplified. Any failure in DNA repair in an environment of permanent cold may be more costly than in temperate environments where the organism has more metabolic resources available for emergency responses. The absence of relevant predators is the third pillar. The evolutionary theory of senescence predicts that longevity is advantageous only when the probability of surviving to advanced ages is high enough. If predators eliminate most individuals while young, investing in maintenance for future decades is an evolutionary waste. The organism probably won't make it there anyway. In the ocean abyss, below 1,000 meters, large predators are rare. Sponges fixed to the substrate have no predators that consume them regularly. Deep-sea corals likewise. Clams buried in cold Arctic sediments are relatively protected. This relative safety from predation creates evolutionary space for longevity investments to be positively selected. and over enough generations, produces the organisms on this list. Depth also matters as an additional factor. Beyond low temperature and few predators, deep waters in many contexts contain less dissolved oxygen than surface waters, a condition called hypoxia. Organisms that evolved in hypoxic environments develop mechanisms for efficient oxygen use that reduce the production of free radicals even during active metabolism. Additionally, high-pressure environments may favor protein configurations that are more stable and resistant to damage. Proteins from deep-sea organisms frequently have structures adapted to function correctly under hundreds of atmospheres of pressure, and those same structures may confer greater resistance to oxidative and thermal damage than proteins from surface organisms. Each of the 6 organisms in this series represents a distinct biological solution to the same fundamental problem: how to persist. The glass sponge opted for a mineral-based architecture that doesn't age the way soft tissue does. The Ming clam reduced its metabolism to the point where aging became biologically irrelevant on a human timescale. The bowhead whale developed DNA repair machinery of exceptional precision. The sea urchin kept telomerase active in all tissues without incurring cancer. The immortal jellyfish acquired the ability to completely restart its developmental program. The Greenland shark simply slowed everything down: growth, movement, metabolism, aging, to rates that make 4 centuries equivalent to decades in terms of accumulated wear. 6 independent solutions. One same result: lasting. What these organisms teach, when considered together, is that human aging biology is not the only possible model, nor even the most efficient one. It is the model that emerged in the lineages that led to land mammals, under the specific selective pressures of that ecological niche. Other niches, especially the cold, deep oceans, produced other solutions, some of which are now the subject of active research in gerontology, oncology, and regenerative medicine. The bowhead whale is teaching oncologists about DNA repair. The sea urchin is teaching gerontologists about telomeres. The jellyfish is teaching developmental biologists about transdifferentiation. The glass sponge is teaching oceanographers about paleoclimate. Organisms that humanity barely knew of decades ago are progressively becoming unlikely guides to questions that human medicine still doesn't know how to answer. There is something deeply human about studying creatures that live for millennia without aging. It isn't just scientific curiosity. It is the projection onto real and measurable organisms of one of the oldest aspirations of the species: to last longer, to decline less, to exist more fully for more time. The difference between that aspiration and the scientific research these organisms inspire is that science is not trying to reproduce the glass sponge's longevity in humans. It's trying to understand the molecular principles that make it possible and to assess, with rigor and caution, whether any of those principles can be adapted in ways that extend human health without introducing equivalent risks. The path between understanding and applying is long, uncertain, and full of obstacles. The starting point, however, is at the bottom of the ocean, where creatures that existed before human writing continue to exist, silently, teaching without knowing they are being studied. The narrative that began with a glass sponge at the bottom of the Indo-Pacific closes a circle that is neither chronological nor geographical. It is conceptual. From the sponge that records the end of the Ice Age in its silica structure to the whale that carries 19th century harpoons as living scars, each organism in this series is simultaneously a living being and an archive. They record in their bodies the time they have traversed. The Ming clam stored 5 centuries of seasonal Atlantic variations in the layers of its shell. The Greenland shark accumulates in its eye lens the isotopic signature of the water it was born in 4 centuries ago. The golden coral preserves in its skeletal protein the oceanographic conditions of 2 millennia. They are living archives and mirrors as well. In studying them, biology reflects back to humanity an image of what is possible when life operates on timescales that human experience will never directly reach, but can understand, learn from, and perhaps, one day, at least in part, imitate.